from Seattle and went to a very nice place called Yorktown, New York, where he became actually my colleague. At the time, we were colleagues from 1985 to 1989 in the IBM Watson Research Center. He then returned in the late 80s to Seattle, where he remains today. Since the early 90s, Professor Tompa took an interest in the area of bioinformatics and became very influential, particularly in the area of sequence analysis and regulatory analysis. And I believe that matter he will speak on today. So without further ado, will you please welcome Professor Tompa. I think it's better to pin it rather than that.
Oops.
This is a wired one.
Okay. Okay.
Stay within this.
I have no choice. Well, thank you very much for that very, very nice introduction. I'd like to also take this opportunity to thank the IMS and the School of Computing here at NUS for inviting me to give this lecture. It was an opportunity that to visit Singapore. It's my first time in Singapore, and I'm enjoying my stay very much. And I want to thank all of you. This is a larger audience than I expected for volunteering to come out in the evening and listen. So I will try to make it interesting. I'm going to talk tonight about, as the title says, the role of mathematics and computer science in recent developments and research in molecular biology. This is an area that is very interesting and also involves the intersection of these three different fields: molecular biology, mathematics, and computer science. And so I'll try to give you a little feeling by running through a few examples of how these areas intersect and how they have an effect on each other. So I'm going to try to do this without assuming any background. So I know some of you come from a background in biology, some come with some background in computer science or mathematics. Probably not too many of you come with a background in all of them. If you have a background in all of them, then you should leave now because you won't learn anything new. So I'll try to make this self-contained, so that you don't need to really know anything about any of those fields in order to get some appreciation for what's happening in this area. So let's begin by talking about DNA, which, as this slide says, I've called it the cells program. So you're all familiar with this picture of DNA, the famous double helix. This picture shows the DNA as though it was spilling out of this cell. Every cell of nearly every living organism contains DNA. And the DNA is, you can think of it very, very simply as being the instructions to the cell that tell the cell how to carry out all of its functions. So all the functions that the cell needs to carry out the functions of life are encoded in the DNA. And we'll talk about that a little bit later today. All you need to know about DNA for today's talk is that, first of all, it's a very, very long molecule, and it's composed of four building blocks, four smaller molecules that are called nucleotides. These nucleotides come in four varieties, which are abbreviated A, C, G, and T. And those four different nucleotides have different chemical properties, which is why they come in four varieties. And it's the sequence of those nucleotides. So this may be a little bit too small for you to see, but each of these little blocks here is one of those nucleotides. And it's the sequence of those four nucleotides that encode the instructions for the cell, telling it how to carry out its processes. Okay, because of this, I'm going to take an overly simplified view of DNA, and this is a reasonably common simplification. I'm going to pretend that DNA is nothing more than just a sequence of those 4 characters, A, C, G, and T, in some order. And again, because it's a long molecule, this is a very long sequence. I'll tell you in just a few moments how long you might expect this to be. So for today, we'll view DNA simply as a sequence over those 4 letters, A, C, G, and T. Okay, so the title of the talk is The Role of Mathematics and Computer Science in Molecular Biology Research, and that suggests that the first question we should ask is, do molecular biologists need any help from computer scientists or mathematicians? So I'm a computer scientist by background, as you heard in the introduction. So I'll try to convince you that the answer is yes, that they do need some help. The reason that I believe molecular biologists need help from some of the quantitative sciences like computer science and mathematics is very simple, and that is that the amount of data that the molecular biologists have to deal with is vast. These systems that they have to deal with are quite huge. And just as the most simple example, let's talk for a moment about genomes. Okay? So what is a genome? This is something that you've read about in the newspaper all the time. A genome is simply the name for the total collection of DNA in one cell of an organism. And so we can ask the question, how many nucleotides are there in a typical genome? What this table shows you is that these genomes are actually very, very large. So for example, let's start with the smallest one in this short table. M. tuberculosis is a bacterium. This is the bacterium that is the cause of the disease tuberculosis in humans. Bacteria are single-celled organisms. These are among the simplest organisms that exist. And even this very simple organism has 4 million nucleotides in its genome. So its genome is a single molecule of DNA, and it's 4 million nucleotides long. So this is quite large. But that's the smallest in this table. If we go to the common fruit fly, its genome has size 200 million. If we talk about any of your genomes in the humans, each of your cell has a genome of size 3 billion nucleotides. So this is enormous. It's huge. But we shouldn't get particularly boastful about this, because there's some very simple organisms, some plants, that have genomes that are much larger than ours even. So this whisk fern, for example, has a genome of size 250 billion nucleotides. Okay, so these numbers alone should convince you that biologists need help from quantitative scientists. Simply to be able to analyze, let's say for humans, to be able to analyze the 3 billion nucleotides, and even before analyzing the 3 billion nucleotides, just to figure out the sequence, what is the exact sequence of 3 billion nucleotides, is a difficult computational task. Now, we should couple this with the fact that what the molecular biologists are trained to do and expert at is what's often called wet lab work. They work in the labs doing experiments at the bench. And they're very good at that wet lab work, but it's costly and very time consuming. So any place where they can get help from quantitative scientists will be very useful. Okay, this is just one example. There are lots and lots of examples of how vast the data is that molecular biologists have to deal with. Here's another view of this same phenomenon. This is a graph. It's titled "The Growth of GenBank." GenBank is one of the public databases that is a repository for biological sequences, molecular sequences, such as protein sequences and DNA sequences. So what does this graph show? On the horizontal axis, we have years from 1982 to 2005, a year ago. And on the vertical axis, here in blue, we have the number of nucleotides of DNA in the billions. And what the blue curve shows here is that over the last, say, 10 or 15 years, there's been an explosive growth in the number of sequences, the number of nucleotides of sequence that have been deposited in this database. So we see, for example, that, let's say, in around 1998, there were about 2 billion nucleotides known of DNA sequences. That was 1998. 7 years later, in 2005, there were 56 billion nucleotides deposited in this database. And there's no sign that this rate is going to slow down. There are, you can see billions of new nucleotides that are being deposited every year, billions of nucleotides of sequence. Okay, so let's talk about this basic question, how to compute the sequence of nucleotides in the human genome. So I mentioned already that the human genome is 3 billion nucleotides long. Each of your cells contains 3 billion nucleotides. And those nucleotide sequences, I should also say, are extremely similar from one human to another. So the genome sequence of my genome and the genome sequence of your genome is nearly identical. It's something like 99.9% identical. So only one nucleotide out of every 1,000 is different in your genome and in mine. So because they're so similar to each other, it makes sense to talk about the human genome. So we can, if we take some human genome and figure out the sequence of 3 billion nucleotides in it, that will be a very good representation of any of our genomes, because they're all so similar to each other. So as I'm sure you read in the newspapers at the time, about 5 years ago, the human genome was completely sequenced, which means simply that scientists figured out the exact sequence of 3 billion nucleotides in each of our genomes. Now, that's a little bit, it's a little bit of a lie or an oversimplification. They declared success in 2001 as though the entire human genome had been sequenced, but in fact, there were certain difficult portions of it, small portions that hadn't been sequenced yet, and those are still being worked out today. I think it's nearly complete now, these difficult sections, or maybe it's in fact entirely complete now. So the first question you might ask is, why would anybody be interested in doing this? Why do we need to know this sequence, the exact sequence of 3 billion nucleotides? If I handed you such a sequence, I said, here's a nice sequence, 3 billion A's, C's, G's, and T's. They come in exactly this order. You might say, well, what am I going to do with this? This is very uninteresting. But this was the first step and a first very important step in understanding how cells work. So remember, I said this DNA sequence contains in it all of the instructions that the cell needs to carry out all of its life functions. And so we can't understand how the cell works until we at least know what do the instructions look like? What is the program for the cell? I'm going to talk a little bit later about why this is only a very first step in this process, but it is a necessary first step. We have to know what does the program look like. Okay. So what I'm going to talk about in the next part of the talk is how this was done. I just want to give you a rough idea of how scientists actually figured out the sequence of 3 billion nucleotides. And I'll talk about it as though we were trying to sequence the human genome, but the same process that I'm describing is the way that we sequence any genome, whether it's bacteria or fruit flies or humans. Okay, so how is this done? We start out by taking multiple identical copies of the human genome. So each of these green lines is intended to represent one copy of the human genome. And I want to stress that they're all identical. So in this picture, I've got six identical copies of the same human genome. So it's easy to do this. We can take some human individual and take a few blood cells and extract the DNA from them, or skin cells and extract the DNA from them. And by taking these cells from the same individual, we're guaranteed that we'll have identical DNA sequences in all of those cells. Right. Now, why do we need multiple identical copies will become clear in a few slides as I explain this process. Now, the next thing that you do is to take these multiple identical copies of the genome and to break them at random places. We want to break them into small fragments, and we want to break them at random places. So we choose these places to break the genomes at random, and we want to do it so that these fragments of the genome are rather small. So the next slide will explain why this is important, but we want these fragments to be random and about of length roughly 1,000 nucleotides long. So remember, we started with genomes of length 3 billion, and we're breaking it up now into quite small pieces, each of length about 1,000. Now, how is this done? You can think of doing this, there are a number of ways to do it. The simplest way to think about it is by a physical process, like putting them into a blender in your kitchen. Imagine you took these multiple copies of the genome, you put them into your blender and turn the blender on, and the blender is just going to cut them at random places. And there are some physical processes done in the lab that are very similar to that. So that's a perfectly good way to think about it. And just like in that blender example, one thing that happens in this process is that the order of the fragments is lost. So they're all mixed up together. I no longer know, for example, that this fragment and this fragment came from the same copy originally, because they're all mixed together. So all these fragments are now mixed together. And not only do I lose the information that two fragments came from the same copy of the genome, but I also lose the information about order. So I lose the information that this fragment comes before this one, which comes before this one. All of that is lost because I've jumbled them all up. Okay, so far, these tasks that I talked about, well, let's see, this task, for example, is done in the lab by biologists. This next task is also done in the lab by a process that was developed by molecular biologists. So what the biologists do next is they take each of these short fragments of length about 1,000, and they can figure out the sequence of those fragments. And here's the reason. Now I can explain to you why they had to break them up into these short fragments. It turns out that the technique that is used for computing the sequence of a fragment only works for very short fragments. So if the fragments are much longer than 1,000 nucleotides, this process doesn't work to figure out the sequence. But for short fragments, 1,000 nucleotides or less, biologists have developed a way to figure out the exact sequence. So I won't explain how that's done. But imagine now that each of these short sequences, we can figure out exactly what the sequence is. Now, what you'll notice is that if I pretend that I know that these two fragments, say, came from two different copies of the genome, and originally they actually overlapped. So they overlapped in the sense that, remember, this sequence is identical to this one, and so two overlapping fragments like this will have some piece that's identical. So in this case, this TC-TTC is identical to the TC-TTC here because they overlap. So that's going to be a key to the next step that we're going to do to try to figure out the sequence of the entire genome. Okay. Now, unfortunately, as I said, those fragments are all mixed up, and so I don't have them nicely lined up like this. So it's very difficult to tell to tell which ones actually overlapped originally. Now, the other thing you have to do, you have to use your imagination, of course. I've written very short sequences here, but imagine that these sequences are each of length about 1,000 long, each of the sequences that I've written out here. Okay. Now, so far, biologists have done this without any help from mathematicians or computer scientists, and it's the next step where we provided some useful service for them. So, this next step now you can probably imagine from everything I've said so far. We now have something that looks a lot like a jigsaw puzzle. We have millions of fragments, each of length about 1,000 long. The biologists, by their process, have told us the nucleotide sequence of each of those millions of fragments. And now we have this jigsaw problem of putting them together, figuring out from their overlaps exactly in what order they fit together. So somewhere in the mix, these are the same fragments that I showed you a moment ago. So somewhere in the mix, we have these fragments, and some of these fragments have a very large overlap. So, for example, if you're imagining these to be 1,000 nucleotides long, you might imagine that 2 of the fragments might overlap by maybe 500 nucleotides. And so we get a very long overlap, such as the example I showed here. Imagine this is an overlap of 500 nucleotides. From those overlaps, we can deduce that these 2 pieces in fact overlapped in the original copies, and we can start to build up by putting together these pieces with large overlap. We can start to build up larger and larger sections of DNA until finally we've reconstructed the entire genome. So this last process is done computationally. So this is the place where computation comes into the picture. The biologists deliver the sequences of these millions of fragments, and then there are computational programs that figure out how they overlap and reconstruct the original sequence. So now you can sort of see the entire process. Why was it important that we started with multiple identical copies of the genome? We need multiple copies because we need these overlapping pieces from 2 different copies in order to assemble the fragments into the whole genome. Why did we want to break them up at random places? Well, again, we needed those random cuts so that we get nice overlapping pieces like this that would allow us to reassemble the whole piece. And I already explained why we needed to fragment them into fragments of length about 1,000, because that's all that the molecular biological technique can handle in the basic sequencing. Okay, so I haven't really addressed the question of where mathematics comes into this process. So in order to try to address that question, let me go back to one thing that I haven't explained yet, which is how many copies of the human genome should we start with when we do this sequencing? So what you can see is that if we started with only 1 copy of the genome, it wouldn't work. If we had only 1 copy, we wouldn't get any overlapping fragments when we split it up. So that won't work. If we start with 100 copies, that may well work, but it might be very expensive. The more copies we start with, the more fragments we end up with, the more sequencing we have to do. And so having lots of copies will probably work, but it makes the process more expensive. And so one of the interesting mathematical questions that comes out of this is, what's the right number of copies of the human genome to begin with? So we'd like to make it small enough so that we'll still succeed. And so let's imagine that we start with k copies of the genome. Then the following is a very interesting mathematical question that comes up. We'd like to ask, starting with k copies and breaking them at random places into fragments of length, say that average 1,000 nucleotides, we'd like to ask, what's the probability that we'll be able to reconstruct the entire genome, or at least reconstruct a very large piece of it? So we want to pick k as small as possible. That will make the process as efficient, as economical as possible, but still large enough that this probability that we can reconstruct the whole genome will be close to 1. Right? So that's one of the interesting mathematical questions that comes up in this process, and in fact, 2 mathematicians Eric Lander and Michael Waterman studied this question and came up with a theory to help answer it. Where does the computer science enter? Well, that's probably clearer from what I said. We need algorithms that are that will do this jigsaw puzzle problem to assemble the fragments into the whole sequence. So that's certainly a computational problem. Now, the process that I described is overly simplified. If things were this easy, we would have finished the human genome long before 2001. There are a number of complications that I haven't dealt with, and I just wanted to give you an idea of what some of those are. So, for example, this lab procedure to sequence an individual fragment of DNA has errors. It will occasionally say some nucleotide is an A when in fact it's a G. So the methods up here have to deal with the occasional error that comes up during sequencing. Something that's even a bigger problem than these small errors are contamination. So it turns out that there can be contamination in the experimental process from bacteria or from yeast or from other agents that are used in the process of doing the sequencing. So we get large regions of contamination that again have to be dealt with in these processes. One of the biggest problems that comes up turns out to be these things called repeats. So it turns out that in the human genome and in many other genomes, there are very large regions of the genome that are repeated multiple times in the genome. So you might have a region of length, perhaps let's say 10,000 nucleotides, that is repeated several times in the genome, almost identically. And now you can see this is going to cause a problem in the sequence assembly. If this fragment here, let's say this fragment here is of length, let's say 1,000, and it's part of some repeat that occurs, say, 10 times in the human genome, then how are we going to know which of the 10 copies of this is the one that intersects with this? It may be that all 10 copies share the same subsequence as this. And so these repeats that are unfortunately very common in the human genome cause all sorts of problems in this process. And again, that has to be dealt with in the computational methods. Okay. So that was just a small overview of how genomes get sequenced. Let me now turn to the question of what is it that we're going to do next. So again, in the newspapers that have covered the Human Genome Project, you probably read around 2001, at the time when this was done, things from newspaper reporters saying, we're done, we're all done. We've sequenced the human genome, it's a success, there's nothing else left to do. And that actually couldn't be farther from the truth. So again, it's easy to imagine why this is far from the truth. Imagine again, I hand you this sequence of 3 billion nucleotides. I say, look, it's a success. This is what your genome looks like. It's these 3 billion nucleotides in this order. You would look at this, you would say, what does it all mean? How can I tell from this what's going on? And that's a question that we're going to be facing for dozens of years to come. The question of what does it mean? What is the program that's encoded in this DNA? How does the cell function, and how does the DNA give it the instructions to carry out its functions, are questions that we're going to be asking for a long, long time. These are not easy to answer. So one of the basic questions that is dealt with in the area called sequence analysis is sort of typical of the questions we're going to be asking for a long time. Now that we have this whole DNA sequence, which portions of it have some biological function? It turns out in the human genome, probably not all of it is functional. There are some pieces of it that are just there perhaps as relics of things from the distant past. but only portions of it have function. And for those portions that are functional, we want to know what is their function? What is it that they do? So really, in the rest of the talk, I'm going to be talking a lot about open questions, things that none of us know how to solve. And that previous one was the first open question. Which regions are functional and what's their function is a giant open problem that scientists are going to be dealing with for a long, long time. All right, so let's come back. In order to talk about functional regions, let's come back to this initial picture that I showed you. We were taking the simplified picture of DNA as just a sequence of A's, C's, G's, and T's. And let's put a little bit of more information into this picture. So some of the functional pieces are the pieces that are called genes. And in this picture, I've denoted some of the genes by these red short fragments. All right, what is a gene? A gene is a portion of the DNA that encodes either a protein or an RNA molecule. So let's forget about RNA for a moment. Just to simplify life, let's talk about proteins. So I'll simplify things and we'll say a gene is a portion of the DNA that encodes a protein. Now, what is a protein? A protein is a molecule in your cell that actually carries out the cellular processes. So DNA actually doesn't. DNA is very inactive. It sits there, it's got the instructions for your cell, but it's the proteins that actually carry out those instructions. They do all sorts of different cellular processes, such as catalyze chemical reactions that are necessary for your cell to, say, turn nutrients into energy. That's carried out by proteins. Proteins transport small molecules such as sugar, transport them around the cell, into the cell and around the cell. They carry signals in and out of the cell so that the cell can tell what are the conditions outside the cell and how should the cell react to those conditions. And they form a lot of the structures of the cell as well. So the proteins are actually the agents that are doing all the hard work, the work that's explained that's given instructions in the DNA. Okay, good. So here we have pictures of little pieces of the DNA called genes. Those genes encode proteins, and the proteins are the molecules that are actually carrying out the cell processes. Okay, so this would be a very important and typical problem of the sort that I was talking about before. Which regions are functional? Well, in this case, what we want to know is where are the genes? So here's the sequence of 3 billion nucleotides. Tell me exactly where are the genes. There's a gene here, there's a gene here, there's a gene here. And once you know where the genes are, we'd like to then ask the more complicated question, what is the protein that's the product of that gene, and what is its function? What does that protein do? Okay, so identifying the genes within the genome is a very good problem. It is largely a computational problem. Again, you can do this in the lab, figure out where the genes are, but because the genome is so large, it would be much more efficient if we could at least try to predict where the genes are computationally and then give those predictions to a biologist to verify them in the lab. It's a good computational problem. It's been studied quite heavily for a number of years. And even with this heavy study, it's really only been partially solved. The best computational methods that we have are probably only about 80 or 90% accurate at predicting genes. So this is still an open problem. I'm going to talk about a different problem, though. I'm going to talk about a problem that's a little bit more difficult than this. Namely, rather than trying to predict the genes themselves, I'm going to talk about the problem of trying to predict those small portions of the DNA that control the genes. And I'll explain exactly what that means. So this control of genes falls under the topic that in biology is called regulation. In the picture that I showed here, this shows that genes get turned into proteins. That process of turning a gene into a protein is something that doesn't happen constantly, but happens only under controlled circumstances. There are certain times and certain places when you want this gene to be turned into its protein product, but you don't want all genes to be turned into protein products all the time. And this question of controlling when the gene gets turned into the protein is what's called regulation. Here are some of the questions that biologists would love to have the answers to, and all of these at the moment in general are completely open questions. So what is the process that turns genes on and turns genes off? And when I say turning a gene on, what I mean is causing the gene to produce its protein product. So what's the mechanical process for doing that? We really don't understand this very well. At what times, under what conditions is a gene turned on and off? In what sorts of cells is a gene turned on? So for example, you have certain genes that perform an important process in your brain, and you don't want those genes to be turned on in your liver because these are genes that have something to do with brain processes. So what cells, what sorts of cells the gene gets turned on in is part of the question of regulation. And then finally, the question is how many copies of the protein get produced. So when you turn a gene on, you don't typically produce just one copy of the protein, but in fact multiple copies of the protein. You need lots of copies of the protein to carry out its function. And so the question of how many copies are produced, at what rate are the proteins produced, is again a question of regulation that we don't know the answer to. So you can see we know very little about regulation. One reason we know so little about regulation is that it's a complicated process, a multi-step process. And I'm going to talk today, in the rest of today's talk, just about the very beginning of this process of regulating genes. So this is a diagram of what the process, what the molecules involved look like at the time when a gene is about to be turned on to express its protein product. So let me introduce you to some of the molecules. So here is a piece of the DNA, and I'm using some of the same colors that I used before. So I've blown up one little piece of the DNA, and here again in red is the portion called the gene that encodes the protein that's about to be produced. Now, nearby in the DNA, near to the gene, are these much shorter pieces that are called regulatory elements. The regulatory elements are little pieces of DNA that play a role in controlling this gene, turning it on and off. And then up here, near the DNA, I've got some proteins. So there are these two proteins that are called transcription factors. They have something to do with regulating this gene. And another protein called RNA polymerase. Okay. Now, what you can see is that I've color-coded these transcription factors to match their regulatory elements. And this is supposed to suggest that this green transcription factor is looking for a target, a small regulatory element where it will bind. So the protein's actually binding to the DNA at that regulatory element. And similarly, the blue transcription factor is looking for this little blue regulatory element where it binds. And once they bind there, together they attract the RNA polymerase. Now this is just an example. Sometimes there may be only one transcription factor that recruits the RNA polymerase. Sometimes there may be 10 or 20 transcription factors, depending on the organism and the gene. But typically there'll be some number of transcription factors that bind to the regulatory elements. They recruit the RNA polymerase. And then there's a long process that goes on, which I won't get into because it doesn't concern us today, by which the RNA polymerase reads across the gene and some things happen and eventually a new protein is produced, the product of that gene. Okay, now the process that I'm going to be interested in is this first process, just the question of where are these regulatory elements that the transcription factors bind to. So remember, the general question we're asking is which portions of the DNA are functional, and the particular functional elements I'm interested in identifying now are these regulatory elements, short sequences where transcription factors bind. All right, so here's our goal for the next little piece of this lecture. We're going to try to identify regulatory elements in DNA sequences. And I've told you almost everything in this slide already. I've told you already the regulatory elements are binding sites for proteins. I said that they're short pieces of DNA. I didn't say how short. They're typically 5 to 25 nucleotides long, so very short pieces. I'm going to think about them as being length 10. That's a typical length, so 10 nucleotides long. And I showed you on the picture that they are typically nearby to the gene that they control. And typically they would be about within 1,000 nucleotides from the beginning of the gene. Sometimes they're farther away, but this will give you a good indication of what we're looking for. Now, the part that is interesting and also makes our problem difficult is this last one. I suggested before that this green transcription factor was looking for a particular regulatory element, a particular sequence of length 10, let's say. But that's a little bit of a lie. That sequence that it's looking for, the transcription factors are actually quite tolerant of small changes to the sequences. So it may be that near one gene, this transcription factor binds to a certain sequence of length 10, and near another gene, it binds to a slightly different sequence of length 10. Similar, but slightly different. So there's some variation in the binding site for this transcription factor. And this makes our problem, the computational problem, interesting and difficult because we're not looking for an exact pattern, but rather only an approximate pattern. It might be slightly different from one instance to the next. So these sorts of inexactly repeating patterns are called motifs in the biology literature, and that's the term that I'll use. So we're trying to find these short motifs in DNA. Now, how are we going to do it? Remember, what I give you is, let's say I have a particular gene of interest. Let's go back to this picture. In fact, let's go back to the picture where all I have is the DNA. So I've got something like 1,000 nucleotides. I know the regulatory elements are somewhere in this 1,000 nucleotide stretch, but how am I going to answer the question of where they are? I mean, these 1,000 nucleotides, they all look the same to us, right? It's A, G, C, T, G, T, G, C, C. It's just a bunch of A's, G's, C's, and T's. How are we going to know which 10 of those are the binding sites for some transcription factor? So in order to answer this, biologists have suggested a very simple but very powerful method. And this method is called phylogenetic footprinting. This is a term that was introduced in a biology paper by Tegel et al. Almost 20 years ago. And the premise that underlies phylogenetic footprinting is this very simple one. Namely, over the course of evolution, functional regions of the DNA tend to mutate at a slower rate than non-functional regions. So that makes sense, right? If anything that's functional in the DNA, if it mutates, it's very likely to be harmful to the organism. The organism it may even be lethal to the organism, and the organism dies off. So there's pressure, evolutionary pressure on the functional regions to stay the same over the course of evolution. The non-functional regions next to these functional regions, it doesn't matter if they accumulate mutates because they have no function. And so what we expect is that over the course of evolution, functional regions are going to evolve slower than non-functional regions. This is the principle that is usually called selective pressure. Okay, so what Tegel et al. Suggested then is, rather than looking only at one organism, let's look at a whole collection of related organisms. So for example, if we're interested in humans, let's look at a collection of mammals. Let's take a collection of different mammals, and from those, let's select the corresponding sequences from different species. So let me give you an example. Suppose we're interested in understanding regulation of the insulin gene in humans. So what we can do is, rather than just looking at the insulin gene in humans, let's look at the insulin gene in humans and cows and dogs and rats and mice and chimps. Pick a bunch of organisms that all have an insulin gene, and let's look at the corresponding regions and now ask the question, which portions of those look like they've been conserved? Which portions look like they've evolved slower than other portions? All right, so consider a set of corresponding sequences from different species, and within those sequences, let's try to identify those small portions of length about 10 nucleotides long that are very similar across the species, much more similar than the other regions. So that's the idea behind phylogenetic footprinting. Now, let's take a look at a small example here. So imagine again that I've got, say, the 1,000 nucleotide region just next to the insulin gene in human, chimp, rabbit, mouse, and rat. So I've written here imagine these are 1,000 nucleotide sequences. And I'm looking for some motif, some piece that looks like it's very well conserved among these. So here's what that piece might look like. I've now shown it in red here. So in human, chimp, and rat, I have this piece that looks like ACGT. Here I've made just length 4 just to keep it nice and small and easy to understand. In mouse and rat, it looks like ACGG. So it's almost the same. Remember, these motifs don't have to be identical. They just have to be similar to each other. And so perhaps this might represent some regulatory element that I'm interested in discovering. And why am I saying that these look like they haven't accumulated too many mutations? Well, if you imagine labeling, so sorry, I didn't explain what's going on over here. Over here we have the evolutionary tree relating these species. So human and chimp are the closest to each other, so they are neighbors in this evolutionary tree. Mouse and rat are neighbors, and then rabbit is slightly more distant, and then back here we have some connection between the primates and these other mammals. Okay, so imagine that this is the evolutionary tree. These nodes that I've marked here in red represent ancestral species that probably no longer exist. They existed in the past. So for example, this species was the common ancestor of mouse and rat that existed millions of years ago. All right, so what you can imagine is that these red motifs here that I show here might have evolved like this. Perhaps in this ancestor, it looked like ACGT, identical to these two. Perhaps in this ancestor, it also looked like ACGT. What we see is somewhere, because it's a T here and a G here, somewhere in the course of evolution, there must have been a mutation. But in fact, one mutation is enough to explain all of the present-day data. So this one mutation from T to G between this ancestor and this ancestor is enough to explain all of the present-day data. Right? With that one mutation, we have ACGG, just like we have in mouse and rat here, and ACGT, just like we have in human and chimp here, and in rabbit here from this ancestor. So with this one mutation, I can explain all of the current instances. So that's what's called in the biology literature, that's called the Parsimony Score. And what we're often looking for in phylogenetic footprinting are sequences that have a very small parsimony score, a small number of mutations that explain the variations from one species to the next. All right. So there have been a number of methods that have been proposed for doing this phylogenetic footprinting. I'm going to show you one that we developed ourselves at the University of Washington. This is work that I did with a former PhD student of mine named Mathieu Blanchette, who is now a professor at McGill University in Montreal. And it was work that we did about four years ago. So this slide and the next slide are very busy slides. There's lots of notation in them, lots of math in the next slide. And I'm not going to try to explain them to you. I'm simply going to show them to you in order to just give you a hint that there is some interesting mathematics going on here. So let me just give you a very broad hint of how this method works that we developed. So again, this part of the picture is very similar to what you saw before. We start with an evolutionary tree and these sequences of length about 1,000 from related species, the corresponding regions. And we're trying to identify what's marked here in red is what we're trying to identify, these short motifs, but we don't know them yet. And so the method it actually proceeds by a classical method called dynamic programming in computer science. And the way it works very roughly is the method proceeds starting from the leaves of this tree, these nodes that represent the current species, and working its way back toward this ancestor, the root of the tree. And what's happening in this process is simply that we're building up tables at each node of the tree. We're building up tables that tell us what is the parsimony score of every possible motif. Okay? And then once we get up to the root, we're going to try to pick those motifs that have very small parsimony scores. Those are going to be the motifs that have been the best conserved over the course of evolution. Okay, so that's all I'm going to tell you about this picture, is just to give you an idea. And here again, just to show you that there's some mathematics involved in here, without giving you any of the details, what this equation represents, this is the heart of the dynamic programming algorithm that I just tried to describe on the previous slide. This equation tells you how to compute the table at one node from the table at its children. And so let me just make this a little bit clearer here. What it says is, if I know the table here and the table here, it tells me how to compute the table at the next node. And then once I know that one and this one, I can compute the next one. So in that way, we're working our way from the leaves up to the root. Okay, and this is the equation. This simple equation, it is actually a very simple equation, tells you exactly how to compute the entries of a table given the entries of the table of its children. Okay, so let me show you a few examples of the application of this. This program we call Footprinter because it does phylogenetic footprinting, and it's actually become a reasonably popular program for doing this sort of phylogenetic footprinting. I'm just going to show you a few of the applications that we did initially to give you an idea of how things look. So one of the genes that we applied Footprinter to was a gene called the beta-actin gene. And this is a gene that's present in many, many vertebrates. So we have here 4 different fish and 1 bird and 5 mammals. And we took the corresponding regions near the beta-actin gene from each of these vertebrates. And here's the evolutionary tree relating them. And ran Footprinter on it, and Footprinter makes certain predictions. It says, here are certain motifs that look like they are very well conserved across all these species. Now, in this is, you can think of this as the output of the Footprinter program. Footprinter would show you all 10 of those species with their motifs, but I didn't have room for them on this slide, so I'm only showing you one of the fishes, chicken and human. And what Footprinter produces at the end, using color and different size characters, it shows you the motifs that it found. So the input, let's say the human input, was this entire sequence, all of these lines of A's, C's, G's, and T's. And similarly for the other species. And what we find are, for example, this red motif. And you can see the red motif in fish, chicken, and human, and also the other 7 species, is very, very similar. So, for example, this looks like CCT, GTA, etc. CCT, GTA, CCT, GTA. So what Footprint is doing is finding these regions that are much more similar to each other than the rest of these characters are to each other. Okay, let me show you a little bit more detail of another gene. This is the CFOS gene, and here we applied it to just these 6 species, pufferfish, chicken, and 4 mammals, pig, mouse, hamster, and human. And here, just schematically, what I'm representing are each of these horizontal lines is intended to represent the input DNA sequence, so a sequence of about 1,000 nucleotides from each of the species. And in color, the different motifs that Footprint have found. So for example, there's a red motif here. I'll show you a little more on the next slide what these motifs actually look like. This red motif, it found in common just to the mammals, pig, mouse, hamster, and human. This blue motif, it found in all 6 species. So one thing Footprinter is capable of doing is finding motifs that occur only in some of the species rather than all of them. So continuing that example, the actual motifs that we found, here are the same color codes. The actual motifs that we found look like this in human. Remember, we talked about Parsimony Score, the number of mutations among the different instances. So the Parsimony Score is given here. The smaller the score, the more similar they are. So for example, this red motif that we looked at on the previous slide that occurs just in the 4 mammals, this red motif actually has a Parsimony Score of 0, meaning it's identical in all 4 mammals. So here we see the score, and some of them have a rather large score, lots of mutations, but that's compensated for by the fact that this is that blue motif that we looked at that occurs in all 6 of the species. So because it occurs in all 6, Footprinter allows a somewhat higher Parsimony Score. These that occur in the, say, just in the 4 mammals, it's looking for a lower Parsimony Score. And finally, in this last column, this just gave us some nice validation of our method. It turns out that some of these motifs, the 5 here where I've written yes, those motifs were already known. So if we looked in the biology literature, we would find that these are known regulatory elements that are known to bind transcription factors that are important in regulating this gene. But these others, 1, 2, 3 of them, were novel motifs. And this is the sort of thing that computational methods are good for. We can propose, by these simple methods, we can propose novel motifs to biologists, which biologists then can go and either verify or show that they're incorrect predictions. And what you can see, for example, this red one that I've been talking about, which is perfectly conserved in all 4 mammals, is one that isn't yet a known motif. So this is probably a good point. Again, coming back to our theme of do biologists need help from mathematicians and computer scientists? I think this is an important point to come back to it. I've been talking as though we as computer scientists can do wonderful things for biologists, but we're very limited. All we can do is make, in this sort of thing, is make predictions. So we have a method that predicts something's going on with these novel motifs, but that's as far as we can go. We now need the biologists to take an interest in our predictions and say, oh, yeah, that looks interesting. I'm now going to go into the lab and try to do the experiments necessary to verify that this is in fact a functional regulatory element in this gene. All right, so I certainly don't mean to imply by any of this talk that we're ever going to replace the biologists. All we're there to do is to help the biologists. Okay, so I'm almost done. I wanted to talk a little bit about future directions and then wrap things up. What other sorts of problems are interesting to biologists and are places where computer scientists and mathematicians might have some role to play? I've talked only about sequence analysis today, but there are lots and lots of other problems. Let me just go through some of these in the list. Genetic networks, this is a fascinating area. The question of which proteins control the regulate other genes. So remember, these transcription factors are themselves proteins, the transcription factors that regulate a gene. They're proteins, they are the product of some other gene. And so you get from this some sort of a network. genes are producing proteins that in turn are regulating other genes. And to understand this, these are what are called genetic networks. To understand this genetic network is critical to understanding how genes are regulated and to answer some of the questions that I showed you earlier. Modeling evolution. This is a very, very important problem that, again, I think mathematicians are going to have a large role in. The question of understanding how does evolution work, how are mutations accumulated over the course of evolution, where are they accumulated? You can see from the discussion about phylogenetic footprinting that those questions have a role in answering questions about current day species. If I want to answer what's functional in humans today, I really need to understand how have mutations been accumulated over the course of evolution. Understanding the function of proteins and RNAs. I didn't talk much about RNAs, but they are other molecules like proteins that carry out the chemical processes in the cells. So understanding the functions of all of these proteins and RNAs is critical to understanding how cells work. Development, how do cells specialize? We talked at one point about how you have brain cells and liver cells. Those all came from the same initial cell. So the question of how do these cells develop and specialize to their specialized processes is a question that we really don't understand very well. Disease models, models of genetic diseases. How can we diagnose diseases and treat them? And that's related to the next question of drug design. Often the way people are thinking about designing drugs these days is by inhibiting certain proteins that are causing problems. So if your body is producing proteins that aren't doing what they should do, or perhaps are doing something they shouldn't do, then one way to design drugs is so that they will interact with those proteins and stop them from the harm that they're causing. And again, designing those drugs is a problem that's going to involve quite a bit of computation. And finally, questions of individualized medicine, which is something that's becoming very important these days. Okay, I think this is almost the last slide. I have one more after this. I want to return again to the question of mathematics and computer science. If you're interested in going into this area, what parts of mathematics and computer science might be relevant? And so here I've listed some of the topics that I think are important to study. Probability and statistics, I hinted at that earlier when we talked about sequencing. Combinatorics, graph theory, information theory, and mathematical modeling. Those are all areas of mathematics that I think are going to have a large role to play here. And then within computer science, there's algorithm design and analysis, databases, because we've got so much data to handle, machine learning methods, and user interfaces. And my last slide is just a pointer. If you want some more information about some of the activities that are going on at the University of Washington in this area, there is a website here on the internet where you can find out a little bit about what's happening in our group. Thanks very much for your attention and staying so late today. No, that's an interesting question. So the question is this whisk fern that I showed in that early table that had a much bigger genome than humans. Why some plants have bigger genomes? The question in general of genome size versus complexity of the organism is something that's, it turns out that there isn't a very close connection between those two. There are organisms, a good example is, well, maybe that one's a good example. The whisk fern, the whisk fern probably, I don't even know very much about it, but it probably doesn't even have many more genes than the humans do. And even the question of how many genes these organisms have isn't a very good measure of the complexity of the organisms. Yeah, so that's right. So the question is whether evolutionary pressure wouldn't drive every organism to get rid of its useless DNA. And that is certainly true to a certain extent. The reason that there is a good reason, for example, why bacteria have much smaller genomes than mammals do. And that's because they have such short life cycles. The life of a bacterium is very short. They produce new bacteria and die, or just split into new bacteria. So they have a very short, what's called cycle time, very short lifetime. And because of that, it's very important that they can replicate their DNA. When they divide, they can replicate their DNA very, very quickly. And so you want to get rid of as much non-functional DNA as possible, because there's competition there. If one bacterium is even 1% more efficient at replicating its DNA than its neighbor, then it's going to take over, and this other one's going to die out because it can't compete. But when you come to something like humans, we have a lifespan of 80 years or 100 years, and so it's much less critical to us that our genomes are compact. We can afford the luxury of duplicating lots of DNA that perhaps isn't useful right now. And you could think of this maybe as playing into the evolutionary future, that all of this DNA that isn't functional now, as I say, it could have been a relic of something that was functional in the past, or it could be something that's on its way to becoming functional in the future. This is sort of the playground where evolution can try out mutations and see if anything is advantageous. Mm-hmm.
So one of the things that computer science usually do is try to come up with approximate answers for such questions.
Right.
Trying to run the algorithm fast but give some approximate solutions. How interested are biologists are they interested in such approximate solutions, or do they always look for exact answers?
Right. Okay. That's a good question. So the question is, if we can't afford to computationally to solve problems exactly, because it would take years of computation time, but we can only approximate the solutions, are those approximations at all useful? And in particular, are biologists interested in such approximations? So this is a very good question. And it's a question that suggests to me that you come from a computer science background rather than a biology background. So mathematicians and computer scientists are very fond of creating a nice, clean, simple mathematical abstraction of what's going on. And I was doing that in all of the talk today. So we're taking a very complicated system, the human genome, human cells, and we're trying to come up with a simple mathematical problem that we can solve in order to understand that. So this requires abstracting away most of the messy detail that's really going on in biology and coming up with a clean problem. Now, the question that you ask is, what if we can't solve that clean problem exactly, but we can only approximate it? And will the biologist be interested? So let me say that the biologist isn't even interested in the clean problem. So this is a very mathematical point of view that you're taking in your question, and it's one that I take as well. The biologists don't care about our clean mathematical problems. They don't care about whether we can solve them exactly or not. They don't care about whether we can solve them efficiently or not. What they care is whether in practice the answers we're giving them look relevant to them, look realistic to them. And so we're constantly doing the sorts of approximations that you say. Most of these problems can't be solved exactly. They can only be solved approximately in a reasonable amount of time. So we're constantly approximating them, and the real test then is when we come back to the biologists and say, here are our predictions, whether they look at them and say, no, that's nonsense, or whether they look at them and say, oh, that's interesting, I'll pursue it in the lab. And so what's critical in this is for us as mathematicians and computer scientists to be in very close contact with biologists, to have a constant dialogue with them so that we understand what's relevant and what's realistic in the problem, and that we don't abstract that away. That's really critical. So a healthy and ongoing dialogue with biologists is critical to this whole process.
Part of my question was what he posed to you, but I would have asked you in a very simple way, in a more biological sense way. And out of your experience, what's the amount of percentage of your predictions has been verified by the biologists?
Good, great question. Great, this is a very good question. So I started out very naively in this when I started working this, and so I would produce predictions like that, the prediction of those regulatory elements that I showed you, and I sort of expected that if I publish these in a journal somewhere, that biologists will leap onto it, that they'll be so interested that they'll leap onto this and they'll immediately try to verify these. Now that turns out not to be the way the world works. So what I've found is that two other things work better, and this is really the sort of work that I try to do. One is producing software like Footprinter that is easy for biologists to use. So the problem is I'm not good at predicting what the right genes are that biologists will be interested in. So the best thing is if I can produce software that's easy for biologists to use, and I think Footprinter is an example of that. Then hopefully biologists will learn how to use it and use it in their own problems. And this has in fact happened. I'm very, very gratified when once in a while I'll see a paper in the biology literature where they say, we used the software produced by our group and it turned out to be useful. The other thing that works much better is if rather than having me pick the gene to do it, if I go to a biologist and say, what genes are you interested in? And so this has also worked very well. I have a number of collaborations with biologists at the University of Washington where I've gone to them and I've said, can I help you with your problem? And they'll say, oh yes, I'm very interested in M. tuberculosis and I'm interested in these genes in M. tuberculosis. Maybe you can help me figure out how they're regulated. And then we'll try to use our tools to do that. And in that case, again, the biologists are very, very motivated because we're now working on exactly the systems that they're interested in understanding.